Ďakujem. Ďakujem pekne za pozvanie, je mi cťou prezentovať na tejto konferencii. Ospravedlňujem sa, budem prezentovať v angličtine, pretože v podstate to je nariadenie firmy a inak sa to nedá, ale ak by boli nejaké otázky, tak ja potom môžem aj v slovenčine povysvetľovať. Right, so switching to English, so accelerating drug discovery and development with artificial intelligence is obviously a huge topic right now, I guess worldwide. There is literally a need to actually accelerate drug discovery. There are not enough options, and those options that are available, especially in innovative medicines, are extremely expensive. So InSilico has a mission to actually try to address this problem, and I will try to present to you how exactly we are looking to do that. First about the company, so InSilico was founded in Baltimore, Maryland, Johns Hopkins University, if you're familiar, back in 2014, after some initial funding provided by the co-founders. The team very quickly recruited many AI scientists worldwide, which sort of created what we call now the AI core. The goal was to look into the pharma traditional R&D and see how we could potentially utilize the, then recently remerged machine learning methods and also invent new methods to address three main problems that we see as challenging. So first of all is the target discovery using omics and text financial data. By target discovery, we mean what is the biological entity in the tissue or a cell that you would like to modify in order to induce some phenotypic effect. De novo drug design, meaning how do you design molecules that are not only active against the target that you found, but also are tolerated, have good toxicology profile, and the patient could benefit from taking them. The third big topic was obviously predicting clinical trial outcomes. You have the target, you have the modulator. How do you make sure that actually this combination passes a very strict clinical trial process? Now, obviously, companies scale up pretty rapidly, raised close to 400 million in capital, which allowed us also to start our own programs. And right now we have one actually entering phase 2 at the end of this month, which is idiopathic pulmonary fibrosis, and we have another 8 that will be going to the clinical testing very soon. A little bit about technology, but looking at the audience, I guess I will very quickly go through. We are not a single AI method company. We literally try to identify the problem, and then we find a method that would be great to address it. And this is just a few examples. But what we are trying to say here is basically InSilico uses a lot of generative approaches. This is something that re-emerged with ChatGPT recently. It was mentioned, but the cool news is that we were using actually generative approach. Back in 2016, we have a number of applications and publications there, and obviously this is just one of the articles written. There's many things the company has done. For example, active compound from inception to validation in mouse models in 45 days. Using AlphaFold to create novel therapeutics in cancer in less than 30 days. We have the lead program in IPF is actually entering phase 2, but also we helped researchers in the field of amyotrophic lateral sclerosis. We were involved in the coronavirus research. We also launched our own fully automated robotic drug discovery laboratory, and obviously there is many more I could be continuing. Now, what is the problem? So this slide summarizes the problem. So basically, it takes more than 10 years. It's close to 12 years now to develop a medicine from the starting point all the way to the end. And it's also very costly. So it's about $2 billion to get that done. The last figure which we could verify was in 2010, 1.8. And we know in the last 13 years, things went up rapidly. So this is just more details about how many steps you need to take in order to actually find a cure and then take it all the way through all these steps while being extremely closely watched by the regulators and always audited by many authorities. Now, essentially, if you break down the problem, it's actually a biology and chemistry problem and the intersection of those, because biology means that the diseases are actually caused in the tissue, in the cell, in different mechanisms, and then obviously what we believe is that if we can apply well-tolerated, low-toxic chemistry into modulating these diseases or cells, then we can actually generate medicines that patients will greatly benefit from. When we talk about target discovery, a little more detail. So how do you find this receptor, the enzyme, the kinase, or whatever other entity that lives in the cell? So our approach is to basically screen all the protein coding genes, which is close to 20,000, and then take a disease of interest. So let's say we did idiopathic pulmonary fibrosis, and then use different scoring mechanism, which we built on deep neural networks to help us score and rank. And then obviously you take the top 10, and then you try to test these hypotheses with the, are they in validation? And then of course you also test them looking at the literature and comparing, which we also do automatically. The next step would be, apologies, it's been jumping a bit. So the next step would be actually the mechanism of action, which is, so once you find this receptor that you would like to modulate, how exactly you are going to modulate it? Is it going to be inhibitor, so are you going to block or deactivate or activate the specific little particle? Now again, once you have that established, we then design molecules that can help you achieve that. And again, we do it extremely quickly and pretty efficiently. This is the system we built. It's end-to-end drug discovery, meaning that Pandomics is actually also available as software, but normally we use it internally for projects. Identifies the targets, also the biomarker for patient stratifications, number of other things. The Chemistry 42 then designs the molecule. InSilico takes the target, the molecule, the mechanism of action, and gives you good understanding of what to expect once you hit the clinical trials. 3 examples how we validated the system is, first of all, is our lead program. This is entering phase 2 now. So we actually took an idea, so like the program inception, all the way to phase 1, which started last February, in less than 30 months for about $5.5 million in total development cost, which, if you compare with traditional pharma, that would be $4.5 million and close to $500 million in getting this drug to the market or to the clinic. The second one, this is done with the collaboration with University of Toronto. We were together researching a disease called hepatocellular cancer. We had access to extremely rare cases and obviously patients and samples. During that research, we stumbled upon a target, which is the cyclin-dependent kinase 20, CDK20. Obviously, there is no structure available about this protein. We don't know how it looks. Nobody ever researched it. So what we did, we tapped into the AlphaFold open source programs, which now anyone can actually access, and predicted the structure of that protein. Once we had a structure, we generated molecules. We then tested them. We got pretty decent figures. I'm not going to bother you now with the nanomolar and micromolar concentrations, but it's pretty active and is an extremely good starting point and probably is going to be in the clinic fairly soon. The third example is, and this is something I discussed with the organizers as well, InSilico built in Suzhou in China, to our knowledge, the first fully automated intelligent drug discovery laboratory. By 6th generation, we mean there is no human in loop, nowhere. Yeah? So it's everything, even the things like automated storage or even transfers of samples or chemicals, all is done by robots. It's connected to the cloud, so you can perform high content screening and imaging, and then obviously different assays, all the way to sequencing by NovaSeq 6000. All the data is instantly uploaded to the cloud, and you can use it. So if you look at it from this perspective, the hospital sits here. So this is the hospital in Suzhou that provides the samples, goes to the robotic laboratory, which processes the samples, uploads the data here. So here we already can analyze the data with Pandomics. Once we identify the target, right away we can deploy chemistry, which identifies or either generates a new compound or identifies existing, goes through the synthesis, and all the way back to testing. Essentially, this will be part of the hospital and will be supporting the precision medicine activities of the hospital. Yeah? So the patient comes and we will be right away able to screen the library and hopefully find and then also synthesize the drug for the patient in days, maybe weeks. And this is just a few thoughts to the end to see what this means, that AI will disrupt drug discovery, faster medicines, cheaper, more options. And obviously, essentially, what we want to do is the precision and personalized medicine, which could be accessible to everyone, because we will make it cheap and we will make it affordable. So this is the goal. Thank you very much. And yeah, thank you.